So, Bismillahirrahmanirrahim. In this chapter, now we are going to discuss the saccadic movement of the eye, a mechanism of successive fixation points. So, what happens actually is that, for example, you're riding in a car and you see a lot of highlights or a lot of street lights, for example, and then your eye it jumps from one light to other light to the next light. So, this jumping takes place at a rate of 2 to 3 jumps per second, and this jumping movement of the eye from one street light to another street light, this is called the saccades. And it is also called optico-kinetic movement. Optico means eye and kinetic is moving movement. So this is optico-kinetic movement or jumping movement or saccade occurs when you are still, or when you are in a moving car, when your body is still, rather you are looking at an object in a motion, like you are riding a car, okay? And in this whole movement, 10% of the time is spent on moving the eye from one object to another, and the remaining 90% of the time is set to fix the eye on that particular object. And what happens during the jumping motion, or 10% of the time, what happens is that the brain suppresses the visual image, so our mind is not consciously aware of our eye moving from one point to another, so that we do not feel nauseous or etc. So the conscious movement from point to point is not actually seen, or the brain suppresses the visual image at that point. Now we will discuss the saccadic movement during reading. In the previous movement, that was the movement of our visual scene. Now we have the movement of our eyes, and the visual scene is stable. Like this book is stable, and your eyes are moving from one word to another to another. So this is the movement of your eyes on the visual scene. So our eyes are trained to do this movement of the on the visual scene from one word to another word to extract the important information. And this movement of the eye is a saccadic movement. But in this moment, what will happen, we are going to do a horizontal movement from one word to the next to the next. But when we are seeing a painting, for example, so how will we scan the painting? We are going to scan the painting in such a way that we are going to scan it upwards first, then we are going to scan it downwards, then we are going to scan it side to side, then also we are going to scan it horizontally, or angulated motion as well, so diagonally as well. But this all movement is not taking place when you are doing the reading, but which means that when you are moving from one direction to another only. So that is why certain people are visual learners, because many portions of their brain are activated when they are watching or looking at a picture. But when they are merely reading a word, then only certain horizontal thing is going to be activated. So that is how saccadic movement are activated during reading. So in the first paragraph, we saw how the eyes were moving, how the eyes were like static and the scene was moving. In the second thing, we saw how the scene was static but the eyes were moving. Now the scene is moving, but the eyes are not moving along with the scene, rather they are fixed on that particular object. So that fixing movement on a particular object, on a particular moving object, this is known as a pursuit movement. And our eyes are going to develop saccades according to the movement of that particular object. For example, you have a systematic movement of one thing that is moving up and down in a wave like motion. Firstly, your eyes won't be able to follow the motion of that particular object, but after a time, if our eyes are going to learn how the object is moving, the brain is going to allow the eyes to form an automatic detection of the eyes, and this automatic detection will become smoother and smoother if that particular object follows the same pattern. So that will result in smoother movements of the eyes, and finally our brain will be able to adhere to the motion of that particular moving object. And this is a cortical mechanism that has been developed to do so. So this degree of automaticity or automatic subconscious control to compute the motion of that particular object for controlling the eye movement by the brain, that is known as fixation movement or pursuit movement. It actually means that your brain follows the pattern, and pattern of a moving object can be recognized by your brain, and then your eyes move according to that particular pattern. So this is how the fixation or the pursuit movement and how the saccades help in this movement. Now we are going to discuss how the superior colliculi are mainly responsible for turning the eyes and heads towards a visual disturbance. What happens is that in your midbrain you have this superior and inferior colliculi, and this superior colliculi is sometimes responsible, and it was, the part of the old system of recognizing objects. So if the visual cortex has been destroyed but the superior colliculus is intact, what will happen in that situation? That, for example, if your eyes are like this is the medial portion, and the lateral portion, if there is some disturbance in the lateral field of vision, your eyes will automatically turn toward that side. But for that thing to take place, the visual cortex is destroyed but the superior colliculus are intact. So what will happen? Anything in the lateral region of the peripheral retinal field of vision, it is mapped by the superior colliculus. That thing goes into the superior colliculus, and the mapping is done over there. There are different layers that are going to map the whole thing out, but this mapping will not be very much accurate as compared to the visual cortex, but there will be certain mapping, and that mapping will send deep or weak signals to the oculomotor nucleus, which is then going to cause the movement of our eye to turn the eyes. So they are saying that superior collicula are mainly responsible for turning the eyes and heads. So this is how, even in the absence of the visual cortex, the superior colliculi can cause our eyes to move towards a visual disturbance in the lateral field of vision, and thus they can cause the oculomotor nucleus to be activated, but the signal is going to be weak because there will be no support from the visual cortex. Now what is going to happen? For example, you have, this is your eye like this, and they are saying that the optic nerve fibers that are going from the eye, they are type M rapidly conducting fibers, and these type M rapidly conducting fibers, they will give a branch. One will go to the visual cortex and other branch will go to the superior colliculus. And what is going to happen? As I told you, the from the eyes there are type M fibers which are going to relay the information, one to the visual cortex, one branch to the superior colliculus, and this superior colliculus, through certain branches of the medial longitudinal fasciculus, it is going to give certain branches to the other regions of the brainstem. And when the branches will be given to the other regions of the brainstem, what is going to happen? That our whole head, even the whole body, will be moved toward the direction of the disturbance. And they are saying that not only the visual disturbances are going to cause this to happen, but there can be certain external disturbances like auditory disturbances, strong sounds, somatic, like a person touching you. So in this situation, similar pattern will be activated through the superior colliculus, and that superior colliculus will send the signals via the medial longitudinal fasciculus to other regions of the brainstem to cause the movement of your head, then your body, towards that specific region. So that is why your superior colliculus have the global role in orienting the eyes, then the head, and then the body toward the source of the external disturbance. But if the superior colliculus is damaged, it is not working properly, your body, your eyes, your head, they won't be able to move towards that particular thing. Now we are going to discuss how the fusion of the visual images from two eyes takes place. For example, they are saying that these are two eyes of yours and they look at one particular object, and the image that is being formed on the retina is on same point, and now this information is going from the optic apparatus through the whole thing into the visual cortex. And in the visual cortex, because they have stimulated the same portions of the retina, that is why their visual signals are going to come onto the same region of the visual cortex. But when they are telling that when your two eyes are looking at one object but the image on the retina is not same, what will happen in that situation? If the image on the retina is not same, that it means that in the lateral geniculate body different layers will be activated. Similarly, these signals are going to travel parallelly to the this cortex, that is the primary visual cortex. Now when they go to the primary visual cortex and the cortex finds out that these two signals are not going to come onto the same region but they are of the same thing, so it will do interference excitation, that is it is going to cause the signals to interfere with each other, and that interference can be constructive, can be destructive. Constructive means to converge, or destructive means to diverge away. These two interference, they can take place to cause the image that is being formed on the retina to be formed at the same place. So the whole point of this interference thing was that the image on the retina is not being formed in the same place, but our body wants that image to be formed in the same place. So there is this interference excitation principle. And whenever the retina, the visual cortex receives the signals of this pen on the same sides of the retina, and hence the parallel fibers are going to excite the same region of the visual cortex, then it means that the fusion of image has taken place and our eyes have been fixed. That is why the signals in the visual cortex then disappear. So the specific interference pattern, interference excitation, it also disappears with that. But to fix our eyes, there is the interference that can be constructive, that can be destructive, it can be converging, it can be diverging. The main purpose of that is to form the image on the retina on the same location, so that the parallel neurons carry it to the same lateral geniculate nucleus layer and it is relayed to the same layer of the primary visual cortex. So our eyes are extremely useful organs because not only they provide the mechanism of looking at objects, but they are going to help us to do stereopsis, that is to judge the distance of visual objects. First of all, we need to know that these eyes of ours are less than or more than 2 inches apart. The distance between them is 2 inches apart, and whenever an object is near to the eyes, that image of the object on the right eye will be formed a little bit towards right side, on the left eye will be formed a little bit towards left side. So it means that they are not activating same regions of the retina. But when the image is very much far away, like the dot is over here, they are going to activate the same regions of the retina. This thing happens. Now they are telling you that the closer the object is, the greater is the disparity. If the object is close to your eyes, the difference on the retina will be greater, whereas the object is far away, the difference on the retina will be smaller. And this difference or this non-register, when an object is forming or activating the same region of the retina on both the eyes, that is known as the object is in register on the retina. But when it is forming different or activating different regions of the retina, it means that the image is not in register. So the greater the closer the object is, the greater is the degree of non-register. So we can get the closer is equal to increased amount of non-register, or they are not activating the same regions of the retina. This degree of the non-register or degree of the difference of image on the retina helps in stereopsis or measuring the distance of the visual images from the eye. And this mechanism is very much a good, or it helps us to distinguish the distance at a distance of 200 feet or 61 meters. And they are saying the neuronal cellular mechanism for stereopsis is based on the fact that some of the fiber pathways from the retina to visual cortex stray 1 to 2 degrees on each side of the central pathway. That the fibers that are traveling from the retina to the visual cortex, they are straying 1 to 2 degrees on each side. For example, these are the fibers and this is the main pathway, but certain fibers, they are straying 1 to 2 degrees away from the main central pathway. Therefore, some of the optic pathway from the two eyes are exactly in registers for objects 2 meters away. Still another pathway is in registers for object 25 meters away. So they are telling you that whenever these fibers are like a little bit away, right? And these fibers are central fibers. When an object is very much far away, like at a distance of 25 meters, it is going to activate these central fibers because the image on the retina will be on the same location, right? But if the object is very much close and it is forming or activating different regions of the retina, so the swaying fibers or the fibers that are 1 to 2 degrees away, they will be activated in such a situation. So different set of pathways are activated, and when different sets of pathway are activated, then the visual cortex gets to know about the degree of perception. For example, the thing that is far away, it is activating central region, whereas the thing that comes closer, it is activating the peripheral region. So your brain is activated by different sets of pathways, and hence it can do the depth perception or distance perception by the means of the region of the brain that is being activated. So that is known as stereopsis or depth perception, or visual image perception, or length perception, or distance perception. You can call it by any name, just to distinguish the distance an object is placed from the eye. So you know what is normal vision? When eyes, they can fix to a same point. But what is strabismus? Strabismus is that you squint eye, or it is also known as cross eye, or lack of the fusion of the eyes on one particular object. The strabismus, it can be different types. It can be a horizontal strabismus, that your eyes are not able to fix on the something horizontally. Then it can be torsional, that is diagonal. Then it can be vertical, one eye is looking upward, other is looking downward. So why does this strabismus, or this moving away or not being able to fix, take place? This often takes place because one eye is, for example, this is the object, and one eye is fixing satisfactorily on the object, but the other eye cannot do it very much greater. Or both the eyes can do the fixation thing, but one eye does it one particular time and the other eye does it another time. So it means that the fusion is done, but it is not a simultaneous fusion. That can also cause strabismus. Soon the patterns of conjugate movement of the eyes become abnormally set, and what will happen in when this thing is abnormally set, then the eyes would not be able to fix on particular object, and they will not be able to activate the same thing on the retina. And when they will not be able to activate the same portion of the retina, then your visual cortex is always trying to activate the interference excitation principle, and the interference excitation principle will never go, you can say, unnoticed, or it will never be suppressed. Now they're saying that what actually happens is that your brain has these two eyes, and these two eyes are going to activate same areas and of the retina, and then the same regions of the visual cortex. But when there is strabismus, that is, there is crossing of the eyes, or there is like the fusion of the eyes is not taking place, then what they do in certain people, that one eye is activated at one time, other eye is activated at another time. But in some people, it becomes a pathology that one eye is activated all the time, and it is going to fix the object or help in fixation, and the other eye is repressed. What will happen in the repressed eye, and what will happen in the dominant eye? In the dominant eye, the neural mechanism, or the fibers that are going from it to the visual cortex, they are very much strongly developed. But in this one, because it is not fixing on a particular object, the pathway is not developed from it to the visual cortex. When the pathway is not developed, then it means it has very low amount of visual acuity. It is 20 by 400. That the thing is that something that this eye is seeing at a distance of 20 meters, a normal person could be able to see that at a distance of 400 meters. So it means that their visual acuity, their strength of vision, their power, it has been decreased so much. It is because that the eye was never used, neuronal connections never developed from it to the visual cortex, and hence the eye was repressed. In such a person, if they are blinded in this dominant eye, then they will have a extreme loss of vision, because this eye has no proper connections to the visual cortex, and hence it cannot activate the visual cortex, and hence it cannot help in viewing the objects. So for neuronal connections to develop, there must be fixing movements of both the eyes that are taking place, so that there are neuronal mechanisms or neuronal circuits being developed. That is so much important. With that, we are done with the fusion of the two eyes. It was a very large topic. I did my very best to explain each and every paragraph to you, because I could never understand that from any video myself. That is why I spent a lot of time on it. I hope you could understand each and everything in detail.